you so much, Marco. Great to be here. Oh, I might need to acknowledge the recording here. Thank you so much, Marco. It's great to be here. They're still working on the microphone here, so I will probably just try to speak loud so we hear it in this room at least, and hopefully...
Fine, if you prefer.
Hopefully the microphone and the computer will pick up this for the recording. Yes, this is going to be a bit about adventures in ligand-gated ion channels. Jean-Pierre did a marvelous introduction to this field yesterday, so I'm actually not going to spend so much time with that. Thank you. So this talk is going to revolve around three main concepts. I'm still fascinated by gating and desensitization in ion, well, membrane proteins in general, ligand-gated ion channels in particular. We're going to talk a lot about allosteric modulation, obviously, given that this is the conference it is. And I hope to have some room for lipid interaction and modulation. But given that we also look into three different channel families, I'm going to need to iterate a little bit over this. So apologies for that. But instead of going through the historical part of allosteric modulation that Jean-Pierre did so amazingly well, I'm going to bring you to the way I was introduced to this field. And that has to do with anesthesia. The person who first introduced me here was Jim Trudell. He died a few years ago, was a professor at Stanford. And when I was a postdoc, he came back and harassing this poor young postdoc that was me, that he wanted to model these channels. And this was, of course, long before Nigel Unwin had the first structure and everything. And I tried to convince the guy that it's completely impossible to do this ab initio. We had a few models anyway. I don't think they were lousy, but I've had the opportunity to come into this field as we got more and more structures, and then holding the hand of somebody who had a very clear target that they wanted to design better anesthetics. There are lots of jokes you can make about Dr. Robert Liston. He's rumored to be responsible for the only surgery with a 300% fatality rate because he happened to cut off the limb of an assistant and somebody in the audience died from shock and everything. But on a more serious note, surgery was horrible before the advance of anesthetics. And we take this for granted, right? That it just works. And the reason you can perform, heart transplant, brain surgery and everything, that you can keep a patient alive for hours on the operating table without the patient screaming. And this is about a 150-year-old revolution started. You can still see the Morton Auditorium if you go to Massachusetts General Hospital, and it's well worth visiting. And the scary part is that it's almost entirely trial and error. Seeing this from an anesthesiologist's point of view, they gradually just tried more and more and more things. There is a famous plot called the MEYER-OVERTON correlation, which pretty much says that the more hydrophobic a molecule is, the better it works as an anesthetic. And look, this is a log plot, right? So if the student were to hand in something like this, where you get a perfect correlation over 5 orders of magnitude, you would assume that the student had cheated. There are a handful of exceptions to this rule with modern anesthetics, but in general, it still holds. If you need surgery, the modern development of anesthetics started sometimes in the 1950s, and then over the decades, there are, well, increasingly better molecules. If you're sedated today, they would likely use either propofol, sevoflurane, or desflurane. But there aren't really any obviously great newer molecules here. During the COVID crisis here, that several hospitals ran out of propofol. Propofol is normally what you would use in the intensive care unit, and it's great because it also induces amnesia so that you don't remember the horrible experiences. But when you run out of propofol, they had to use older anesthetics, and that led to several patients having really horrible psychological side effects of being sedated for weeks. The other challenge here is, unfortunately, that desflurane in particular is a very horrible greenhouse effect gas. So Scotland is outlawing desflurane from later this year. And there is, I think there is legislation in the process of outlawing desflurane from all of the European Union from 2026, which pretty much means we're down to sevoflurane. So that we would like better compounds, but of course, anything that should not be a greenhouse gas can be full of fluorinated or brominated atoms. The way these works, we know better now. We know that they don't work on the membrane. That was thought for a long time. And I won't have time to go through everything here, but Jean-Pierre and other speakers have shown that these channels, the ligand-gated ion channels in particular, they sit in the postsynaptic membrane, and they basically influence signaling in two ways. First, you have receptors like the acetylcholine receptors, where we can inhibit the excitation. So this is an excitatory synapse. Or you have an inhibitory synapse, where you instead can have the same effect by potentiating the inhibition. And if you start to think that that is complicated and many things, then just wait. This is going to get much worse. So already now, and surprisingly, these common anesthetics have both effects. And I have no idea why evolutionary, they inhibit one class of channels, they potentiate the other, but since these two channels have the opposite, they conduct the opposite type of ions, right? They still have the same functional effects, which is a bit absurd. The other complication that Jean-Pierre alluded to, that these channels, they don't only have two states. It's well known experimentally that if you have an agonist or something, you open the channel, but then the effect gradually decays. They fall into some desensitized state that is non-conducting, but you can't really activate it directly again from that state, which functionally makes a lot of sense, right? If you don't want the doors to the cell to be wide open, so you want an agonist bind to induce a short opening, and then you want the channel to desensitize, let the agonist to dissociate, and then you're back to the relaxed state. But understanding how the desensitization process works, it's a completely separate part from the normal gating. There are numerous channels known here, and I won't go through this in detail, but over the last 15 years or so, a number of groups, in particular at Pasteur, have been able to unravel one structure after another of these channels, partly by using prokaryotic systems, which is a separate story in itself, because prokaryotes obviously don't have a well-developed nervous system, but they're amazingly good model systems. And this is roughly where I got interested in the field properly, since Pierre-Jean and others had beautiful effects where they saw alcohols binding in particular sites in these channels, while we had other results. So basically, there are two binding sites in these channels, and the big question at the time was, is it the site inside each subunit that's mostly responsible for these potentiating effects, or are there sites between the subunits? I won't have time to cover that story in detail, but you can mutate the bacterial channels to make them behave much more like human channels. And the cool end result there is, in a way, we were both right and both wrong, because these channels, they bind things both inside subunits, they bind things between subunits, and they also bind things in the pore. And if I had to make a bet today, caveats, I would say that both of those binding sites at the top potentiate the channels, while the inhibitor effect is mostly things binding as a plug in this pore. And we've done a bunch of beautiful studies, in particular thanks to Marc Delarue, where we've been able to show this. The problem there, though, is that what happens if one compound here binds to all these sites? Is it going to potentiate, or is it going to have it be a positive allosteric modulator potentiate the channel, or a negative allosteric modulator inhibit the channel? And then turns out you're going to have both effects. So if you play around with these binding sites and mutate them a bit, normally this channel GLIC in the particular case, the wild type of this channel would be inhibited by propofol, so that it's these bars here. The more propofol you have, the more inhibited it is. Works perfectly fine. If you instead look at the purple bars though, for the tryptophan mutant, what you see here is that when you go from 1 to 10 to 30 macromolar propofol, it potentiates the channel. And then somehow the effect starts to decay at 100 micromolar propofol. And suddenly at 300 micromolar propofol, not only has the effect decayed, you have the opposite effect. So the compound that was a positive allosteric modulator at 10 to 30 macromolar is suddenly a negative allosteric modulator at 10 times higher concentration. So it's not just that it depends on the channel. It's not that it depends on the specific modulator. It's not that it depends on the mutant. It depends on the concentration of the compound. So that, everything we all learned on the undergrad level about reaction, it's wrong. It doesn't work. There is nothing here that says that things are going to change. The arrows will change, that completely change direction depending on the concentration of the compound. We have understood this a bit, partly by doing both simulations and electrophysiology, that you can actually influence both the depth of these sites by literally mutating the binding sites. And in particular, you can influence the accessibility of the binding sites. And now we started getting into K on and K off, right, that we spoke about yesterday. It's not just a matter of the binding coefficient, but also realistically, how slow will these effects happen relative to, say, the desensitization? Because now you have three timescales, right? The timescale of opening, the timescale of desensitization, and the timescale with which our allosteric modulators bind. And I won't have time to go into lots of details here too, but Stephanie, who was an amazingly skilled graduate student in the lab at the time, she kept making these mutations, and then she kept sighing that there was something wrong with our perfusion system and the electrophysiology because it must have been contaminated. Because no matter what happened here, that normally this is fairly quick effects, and after 30 seconds washout or so, the effect would have disappeared. But for some of her mutants, you could take half a minute, 1 minute, 3 minutes, up to a quarter or so. And if you had applied something that, even if you applied something for 3 minutes here and then you wait 10 minutes, somehow there's a memory effect. And obviously that's an effect that if you would have some contamination in the perfusion system or something, so that we keep adding the allosteric modulators even when they're not there. Now, once we have replaced that perfusion system 2 times, we actually realized that what had happened here is that we have influenced the accessibility to these binding sites. So you have the allosteric modulators binding super hard. And they can bind like for 30 minutes or an hour. But note that the allosteric modulator per se will not open the channel. So what happens here is that you have the allosteric modulators that are highly hydrophobic. They go into the membrane partition. They are able to enter these pockets that are now much more accessible. They bind super hard in the allosteric modulator binding pockets, and then they stay there. And absolutely nothing happens until you also add the agonist. And in this particular case, it's a pH-gated channel. And then, boom, you have a much, much, much stronger effect. And I'm not sure if that conclusively explains the MEYER-OVERTON hypothesis, but it really emphasizes this effect. The reason why the MEYER-OVERTON correlation works is that all these super hydrophobic compounds, they work because they need to go into the membrane first, and then they access these binding sites through the membrane. And obviously, if they're not hydrophobic, no matter how good they in theory would be at that binding site, they would never be able to access the binding sites. So somewhere there, I swore that we would only do simple electrophysiology to support our simulations. And then we got a CRYO-EM facility in Stockholm, and I had some great students who started doing computational CRYO-EM work. And then suddenly I had a student who was confident that she was super interested in these channels, but she was an experimental student, so she wanted to determine CRYO-EM structures of these channels. And I'm not quite sure what happened. I sighed that it's really fun to learn new things. I'm not sure. Incompetence is probably not the right word, or it is, but I really love this process, taking a student by the hand and exploring a new field together. So we started exploring CRYO-EM. And so since a few years, we're apparently a structural biology group. I wouldn't call myself that, but we're classified as structural biology in Sweden. So we now have structures of multiple states of these channels. And as a simulation person, it's great that you can suddenly target and determine structures specifically of the channels I can simulate, rather than just a much more complicated experimental system. What we saw from these structures is that, and I'm skipping part of the simulations here in the interest of time, is that for virtually all these channels, the normal resting state structures that you have on the left here, they are characterized that they're quite low resolution in CRYO-EM, and they're also very floppy, flexible from a simulation point of view. Ilpo mentioned that a little bit yesterday, that in many cases, you might not even have one well-defined structure, depending on how you define structure. It's really an ensemble of structure, which also makes sense, because those of you are physicists means that if you have an ensemble of structures, that means that you have great entropy, and great entropy is going to give you a low free energy. What then seems to happen is these undergoes first intermediate states and then end up at pH 4, 3 or 4 when they are actually activated. These structures are much more well-defined. It's not a single perfect state, but it's a much more narrow distribution. And what likely happens here is that at activation conditions, you have a much narrower, much well-defined, more well-defined structure. The question, is it stable? Yes, of course. In some sense, it has to be stable. Otherwise, we would not see them in the experiment. But they're likely not as stable because this really it's a transient structure that is open for a few milliseconds or something, and then they will desensitize. And in this case, we don't even have any proper desensitized structures. Now, that leads into this entire desensitization process, right? And if we would like to model this either in an experiment or computation, we have to compare this a little bit how the channels actually work. So if we were to do single channel patch clamp recording, which we are so avoiding, we're not doing that. You could actually literally track a single channel, whether it's open or closed. And if this is then also as the desensitized states, you should be able to, classify in a simulation, in a particular, are we right now in a closed state? What is the likelihood that if you are in the closed state, if you would transition to an open state? If you are in the open state, what is the likelihood that you would transition to desensitized states, etc.? This works great if you could simulate things for a millisecond. We can simulate things for a millisecond. There are a couple of tricks, though, that we used heavily when I was at Stanford, and Vijay Pandem, I think we worked a lot on folding at home together, small globular proteins trying to fold them. These are far more challenging systems and far longer timescales. But Catherine and the team had this idea that in principle, we should be able to probe dynamics on millisecond scale using Markov state models. And in this case, we now have both open and closed structures, right? Experimental structures, and they might not be perfect, but they're decent approximations. Then we can interpolate between these structures, and possibly quite horrible interpolations. All these interpolated structures we can map back to atomistic, put them in a membrane, and then start running simulations. Hundreds of simulations, thousands of simulations, so that you have maybe 1,000 simulations, each of the length of a microsecond or something, and then just use this to sample the entire landscape. Then we need to apply a bunch of mathematical tricks, if you call it here, because there are strong correlations in an individual simulation, but really detect, has things moved from, say, an open state to a desensitized state? Or sorry, open and closed in this case. And I was still, when I saw the first results here, I wasn't quite sure whether part of me was excited, because at pH 7, when these channels are supposed to be closed, having a beautiful results. These are just simple schematic energy landscapes. And the blue basin here is the lowest free energy, and all these structures are the experimental closed structures. So we definitely predict that under resting conditions, by far the most stable states are the closed states, and there isn't even any local minimum at the open state structures. That's great. And then you drop the pH to pH 4 when things are supposed to be open. And then I started to scratch my head. Sure, you have a local minimum here. We so hope these two wells should trade place, right? At pH 7, pH 4, you would now expect the open states to be more stable. And then I realized that I had my physicist license jacked. This is embarrassing. So thank you. I was wrong. Catherine was right here. We don't have open probabilities for GLIC, but for a few others of channels, ELIC and others, you can actually calculate the conductance. And the open probability for most of these channels, for an individual channel to be open and calculated, is in the ballpark of 10% or so. So even under activating condition, 90% of the channels will be closed and roughly 10% of them will be open. And anybody who has studied physicists and the Boltzmann distribution knows that that means that this state should still be the most stable one, while you should have a small population here. And I think if you do the math here, we end up with 8% open channels, which is probably just luck, I think. But the point is that even at activating conditions, the activated state is never going to be the most stable one. If it was, you would have more than 50% open probability. The cool thing that, of course, when you submit this, you're always asked, yeah, but you might have been lucky there. Can you reproduce mutational effects? And I think we can. So for instance, we can take the pore here and replace an isoleucine residue to make the pore slightly more hydrophilic. And in that case, we definitely increase the open probability of the systems. Or we can use another mutation, H235Q, that Mark in particular studied in detail. And we know that this mutation slows down the transitions between open and closed state. And here too, we appear to have a significantly higher barrier when we introduce them. Now, mind you, this is still a very simple system, but somewhere here we start to get excited. We might actually be able to use simulations, not just to roughly understand how a channel is moving, not just to understand the binding affinity, because that's the other problem, right? It doesn't help me to predict, say with free energy calculation, how strong something is binding just in the closed state or the active. Everything is about this balance. How much do you influence the open state relative to the closed state? And do you actually alter the distributions between these two? So you can start doing lots of more realistic models for PLG gating here and everything, but I'm going to move on to the next slide here. So with Markov state model, you could just simply tell the computer to classify things just in two states. What is more similar to open and what is more similar to closed? And once you do that, we can use the Markov state model to actually deduce the kinetics. That is, how fast do we expect these transitions to be? And what we always see here is that transition from open to closed are always very fast, but actually opening the channel takes in the ballpark of 10 or 15 microseconds, or it's probably decent. Again, it's hard to make any kinetic measurements directly on GLIC. But there is no reason we have to force this to just be 2 states. You could try to classify this in more states and see what happens if you use 3, 4, or even 5 states. And once we started classifying this up to 5 states, we started, what you see up here in dark blue is some sort of secondary closed states that we're not quite sure what it was at the time. But we didn't dare to stick our neck too far out in the paper, but we might actually be beginning to start to see desensitization. The problem with GLIC, though, is that the entire desensitization process is complicated. It's also a bit unclear whether the experimental structure is open or desensitized, but I'm going to come back to that. We published this, but then Urska, this other student, had determined all these structures. We had a problem in the structural biology too, because there again with GLIC, we saw some semi-open states or so, and we were not quite sure. We argue, of course, that the pore is open, but we're not really confident whether we saw open states, and it's really difficult to classify things in cryo-EM. But what we had seen in the simulation, Catherine was quite confident that she saw lipid binding. So we redid all these Markov state modeling, but now classifying things not based on the channels, but based on the lipids. And the cool thing is that you can do this both in this, if you start doing this in the simulations, and this correlate this with your cryo-EM densities, the simulations help us resolve lipids in the cryo-EM densities. And not only do they resolve lipid densities in the cryo-EM densities, but they appear to be state specific. So depending on whether we have open or closed channels, we have slightly different lipid binding. Now, mind you, that I would actually argue that 2 of the binding sites, to the leftmost and the rightmost, they're roughly the same, but we have a second site in the middle that might not be part of the resting thing. We can classify this based on how closely the lipid are interacting with the channels, but the net take-home effect, and I think this is hopefully going to appear in eLife in a week or 2. But I think the net effect of this, that Catherine is predicting, that I think, at least for us, this is the first example we've managed to classify new open states that are resolved in cryo-EM, and they're definitely resolved from the cryo-EM. But we somehow used the simulations to find them. And there is no question that this is an experimental structure. It's deposited in the PDB, and they've accepted it as an experimental structure. But it is an experimental structure that was guided by a simulation, which has made, at least to my field, is the first time we've done it. And of course, I would say I'm probably 75% confident that we have this one specific lipid binding site between the 2 subunits that is likely only there in the open state. So we might have lipids directly binding and stabilizing the open states, which is interesting. That in the prokaryotes, it's one thing, but in eukaryotes, a bunch of these channels, they do not work particularly well unless you have cholesterol in the membrane. And it has been an elusive project trying to find these cholesterol binding sites that I'm going to come back to in a few seconds. Now, as fun as those prokaryotic channels are, they are not super important to the pharmaceutical industry for obvious reasons. Though over the years, we've gradually started to dip our toes more into eukaryotic systems. And this would not have been possible without a beautiful collaboration with Ryan Hibbs in first at UT Southwestern and now at UCSD, that initially were responsible for all the structures here, and then they've gradually helped us to determine our own eukaryotic structures that I probably won't have time to tell you about today. Jean-Pierre told you lots of things about the nicotinic acetylcholine receptors, and I'm actually going to start by talking a little about the heteropentameric one, because this is also a key difference to the prokaryotic systems, right? In eukaryotes, not only does it matter that you have different subunits, but the specific order of these subunits and the binding sites between them matter tremendously. One of these systems that we started out to study is a ganglionic receptor, alpha-3 beta-4. This is remarkably important in the autonomous nervous system and everything, but it's also a major target of all types of anti-addiction drugs. The canonical agonist here is obviously acetylcholine. It's a nicotinic receptor, so you can also have nicotine binding. One of these drugs that is being explored is a drug called AT1001. Exactly what it does, it's not super important. But the idea here is that we wanted to find out, why is this drug so specific to that type of receptor? Oh, sorry, I forgot to tell you that. But when it says alpha-3 beta-4, you might have drawn the conclusion that there are other receptors, and there are indeed. So another important receptor is alpha-4 beta-2. And this drug, AT1001, is highly specific to this type of receptor only. So it binds alpha-3 beta-4, but it does not bind to alpha-4 beta-2. And that's just two of the receptors, right? There are dozens of these receptors, and everything in pharmacology, it's not that hard to find allosteric modulators to them. Pretty much anything is an allosteric modulator, I feel sometimes. The hard part in all types of drug design, can you find an allosteric modulator that specifically targets one type of receptor you're after, but not the other ones? And that's much harder. So Anand here in Ryan's group determined beautiful structures of this in nanodiscs, and it's one of the first examples. We have not proper cholesterol bound, but we have cholesterol hemisuccinate bound in the transmembrane domain, which is pretty cool. Remember the cholesterol importance I told you about? We also have beautiful bound structures here. And these structures we've been able to simulate quite a lot. So if you compare on the top here, you have nicotine bound, and the normal alpha-4 beta-2 receptor would be the one in gray here. And you see that the alpha-4 beta-2 has a much smaller pocket. So in alpha-4 beta-2, nicotine pretty much exactly matches the size of this pocket, while in alpha-3 beta-4, this pocket is a bit too large. It doesn't quite fit. It still activates the receptor, but it's not that great. Now, if we instead take this AT1001 drug, do you see that this drug is significantly larger, right? So this drug fills out this binding site much better. It really activates this receptor, but it doesn't really fit in the binding sites above. And that's also a concept that will come back in a few of these structures. It's very delicate and typically has to do with the size of these binding sites. In this case, it's an agonist binding site in the extracellular domain of the receptors. Add one residue in these loops or change 2 or 3 residues, you're completely going to change the properties of these binding sites. It could be that it's a positive allosteric modulator to activate it, or it could just be that you want to inhibit something by binding in this very specific site. And that's kind of what AT1001 is doing. Jens had a beautiful talk about agonists and antagonists yesterday. So let's see if I get this right. AT1001 is a partial agonist. That sounds strange. That's not going to inhibit the channel. Well, but by being a partial agonist, it induces part of the motion that would normally open the channel. But what this appears to do, it makes the channel desensitize faster. And when it desensitizes faster, it is suddenly no longer conducting. So you have a partial agonist that end up having the effect of an antagonist. And it just keeps getting more complicated, right? That how on earth would I predict that with a free energy calculation? It's something binding in a site. You would have to somehow predict the effect this would have on the entire rotation on the domain, right? And everything. Binding is not going to tell me anything. I will not be able to tell you how fast this will make the channel desensitized based on how strongly it binds. Which is, I'm not sure, it's a mixture of fascinating and scary. We see this as simulations too, but we're running out of time here a little bit, so I will continue. This led us to the next project, because in parallel to this, we were also working with Colleen of Yellow, and Colleen, a few years ago, published a beautiful structure of alpha-7, which is a homopentameric acetylcholine receptor, but I think this was the first example ever where we got a single receptor structure in 3 different states. So we have it on the resting state, or it's mostly resting, but it's bound to an inhibitor. We have it in a state that is likely desensitized. So epibatidine is an agonist, but the 4 here is definitely closed. And then we have some sort of semi-strained super open state where we have both epibatidine and agonist, and a positive allosteric modulator called PNU bound. You can see exactly how large the 4 is there. One question here, though, that Colleen had a huge problem with when they were submitting this is arguing with, is this actually an open state structure, or has something happened with the structure in the middle? Alpha 7 is interesting also because it's specifically selective to calcium ions. So what Yushen and my team started doing, sadly he's going to graduate this fall, but it's been amazing in all these collaborations. He first started doing atomistic calculations and really calculating what is the conductor, what is the free energy in the pore here for different type of ions. And you can actually use MD simulations quite efficiently to classify these structures, and these are not expensive simulations. So first, in the inhibited structure, it's easy, super high barriers, everything is closed. In the activated channels, it's not good to conduct chloride, which is good, we should not be conducting chloride, while both sodium and in particular calcium have much lower free energies. There might be slightly large barriers here to calcium, but they are definitely corresponding. We're talking about a few kilocalories per mole or so. So we're definitely have a conducting channel there. And finally, in this desensitized state structure, at first sight, these curves might seem roughly the same, but you see that we have like a 5 times larger free energy difference here. Here we're going to have differences between the lowest and highest peaks of 10 to 15 kcal, and effectively that's a non-conducting structure. Now, that's cool in some ways. The only problem is that can we actually translate that to conductance? Because that's a single atom, single ion we're pulling through. So what Yushen did again, and I think Jean-Pierre mentioned studies by Marco, right, that you've done computational electrophysiology. We can actually mimic the EFIS experiment in the computer here, just by copying this channel to turning it upside down so that you have 2 wells here, one with high and one with low ion concentration. And then we're just calculating the flux of different ions as a function of the charge across the membranes here. So you can quite directly calculate conductance curves here, and then we end up with conductances here in the open channels that in the ballpark of 200 picosiemens or so, which is pretty much right on spot where we would expect them to be experimentally too. And you might think that I am a computational person in many ways, but over the years, we've burnt our fingers a couple of times. I've always been very cautious about simulations. I think that it's a great Gedanken experiment, but you can't really get anything quantitative from simulations. I think I'm too conservative there. And I'm so impressed by many other groups. And I think we too are increasingly sticking out our neck here. Modern simulations are quite predictive, not for all properties, mind you. There are still landmines here. But for many of these properties, simulations are excellent at reproducing both electrophysiology and experimental binding. Now, there was only one problem with those experimental structures in that superopen state, though. We could not see this positive allosteric modulator in the CRYO-EM density. Nothing whatsoever. And of course, it has to be bound, right? It had a clear functional effect on the structure, but we can't see it. No idea where it's bound. So for the first time, we started doing, or Yushen started doing proper coarse-grained simulations. That normally you would have a simulation just studying the protein in atomistic detail, and we're talking about a few microseconds here. Ilpo and others have long done amazing coarse-grained simulations, and suddenly Yushen started doing, 4 or 5 simulations, 50 microseconds each, and then assemble realistic composition membranes that we call them brain lipids. It's not really from brain, but it's a composition that corresponds to roughly what you would have in the brain. There are a couple of interesting results here. First, we see that the open states appear to compress the membrane. That could be an artifact, mind you, and it's quite possible that it is an artifact that can't be observed. But another potentially interesting explanation could be that the membrane acts more or less as a springboard. Remember that this open state should only be transient, right? So that if you're moving to a state, you're pushing out. But of course, when you're pushing out, the lipids are actually pushing back. And this might very well contribute to this effect that the open state quite quickly relaxes to something else where the pore is closed. The other thing Yushen found is that we appear to have quite different interactions of the cholesterol. All these cholesterol molecules, we didn't place them anywhere. They were just out in the membrane, but they diffuse and bind both between the subunits and close to this MX lattice, where we have both in the resting and open states, they're roughly similar, while particularly in the desensitized states, the cholesterols are binding further up. Now, at the time, we didn't think too much of this. We published this, but the cool thing is that a few months after our paper was out, Shao published another structure where they saw cholesterol bound in specific the place where we had predicted it. This would not have been cool had we not published our paper 2 or 3 months before theirs. And again, had you asked me 5 years ago, I would say coarse-grained simulation, yeah, fun to look at, but you can't really trust them. Well, I'm not so sure about that. So we're doing a whole lot more coarse-grained simulations today. So the other question then is, can we find this PNU binding site? Functionally, it has a super strong effect. So, well, let's try to add some PNU molecules. In hindsight, we should have added more than 10. But it's also good, if you only had 10 PNU molecules in this gigantic membrane, the likelihood that they're going to end up in the right site by chance is virtually nil, right? And I'm going to start that movie. And we don't really get any significant density in the inhibitory states, resting states. In the activating state, we see clear, beautiful PNU density between all these 5 subunits. Yes, you see that the PNU starts to bind there. And you're going to see it flipping between a few states. And then when you move over to the desensitized states again, we mostly abolish the binding. There's still a little bit of PNU binding there, but not as much. And this works great on atomistic scale too, if we backmap it. That could, of course, have been an artifact, or we could have been lucky, right? Well, there is an experimental way to test this. There is a mutant known to inhibit PNU binding, if we're M253L. And if we do this in a coarse-grained simulation, boom, we've abolished the binding. Yeah. So I think we have a decent mechanism of alpha-7 gating and modulation, including all 3 states here, including both the activation and desensitization process. But since we only have 5 minutes, I'm going to need to continue a little bit on some unpublished stuff. Remember how I said that we could do Markov states models when we had an open and a closed states? Now we have an open and a closed and a desensitized states. So in principle, we can do the same thing. We can interpolate between all these 3 structures. Now, desensitization is a much slower process. So this was kind of a Hail Mary shot. But Hail Mary shots occasionally work. So this is based on roughly half a millisecond of simulations collected. And we see beautiful transitions all the way between open, closed, desensitized. And then we also appear to have wells at a few intermediate states. And these intermediate states are interesting because first, there has been a discussion in the experimental literature that there might actually be a pre-open state that we call flipped. That likely corresponds to the state, metastable intermediate. And in terms of the desensitized state, there are potentially 2 desensitized states for these channel 2, exactly why I won't have time to go into, but different allosteric modulators can force it into either of these states. And I'm going to come back to that in a second, but given a Markov states model, we can start to calculate the timescales here, how long it would take to go between these different sets. And these are just numbers, right? So what should we compare them to? Well, for eukaryotic channels, there is a lot of more if it's done. Opening for the NACR should be in the ballpark of 100 microseconds. And that's what we have roughly here, right? You stay open for in the ballpark of 100 microseconds. And in this case, the fastest way out of the open state, I said you might fall back into flip states. Again, there might be a bit luckier that, think about this as order of magnitude estimates. But I think we're on our way to actually sample kinetics of actual eukaryotic channels, not just opening, but also desensiting. And you can visualize this now by stitching these simulations together. So here we are in an open state, and then we desensitize, we move over to the intermediate, we move over to the closed and flipped. And at the same time here, you can see what part of the simulations these correspond to. I'm not going to go through the entire millisecond simulation here. The problem is, of course, we're going to spend most of the time here either in closed or desensitized states. But given this, we can start at what happens if we add cholesterol. Remember what I said, cholesterol is necessary for these channels to work, right? There are also indications that cholesterols might actually somewhat stabilize the desensitized states. And that appears to be exactly what's happening. When we add cholesterol to the system, and again, we're not prescribing any binding sites, we just have it in the membrane, cholesterol stabilizes the desensitized states. We are almost out of time there, so I'm not going to go through all my last 9 slides here, but we've also played around quite a lot with allosteric modulation in the GABA receptors, and I'm going to spend just 3 minutes on that to take you back to the anesthetics here. We worked a lot on trying to understand, in particular, how anesthetics and benzodiazepines bind, because that is, to the best of our knowledge, very similar to how the alcohols bind. There are a ton of binding sites here, and we experimentally believe to have binding sites both extracellularly and in the transmembrane domain. DCP here is a diazepam, which you probably know better as Valium, while FLM is flumazenil, which is the classical antidote to Valium in the emergency room. And together with John Kim in Ryan's lab here, we've determined structures of these channels. And both of the phenobarbital and classical anesthetics here, etomidate and propofol, we see them binding beautifully in the transmembrane domain. But you also here see that the binding here is quite specific to subunit interfaces. So again, the subunit composition is critical for these channels. And if we start with the wild type here, wild type GABA, it's a fairly, I was about to say boring structure. It's not boring, it's a beautiful structure. But if you take this GABA structure and either simulate it, it's going to behave roughly like a normal membrane protein. Then we can add diazepam to it, again, Valium. What diazepam appears to do is that it really tightens up the structure and enforces this compression that, in this case, directly of the transmembrane domain and the rotation strongly associated with opening the channel. So here we definitely see that diazepam acts as a positive allosteric modulator and in reducing the opening process. Or we can add flumazenil. And if we add flumazenil, and we see this clearly both in simulation and experiment, we get the opposite effect. So flumazenil displaces diazepam and has the opposite effect of this, that loosening up the channel a bit, the subunits are not interacting as closely, and then we don't get the positive allosteric modulation effect. And it appears that we can explain this already by, normally you would have diazepam bound both in the extracellular domain and the transmembrane domain. And that's also a common characteristic, right? Multiple binding sites. But what we can do in the simulation is let's start by displacing it just in the extracellular domain while we allow diazepam to stay bound in the transmembrane domain. Because diazepam should be able to displace, flumazenil should be able to displace diazepam. And this is actually what happens in simulations. So that when you're adding that flumazenil in the extracellular domain only, about a microsecond later or so, we see the diazepam in the transmembrane domain leaving. And that's why you end up with the structure that you only have flumazenil bound and there is no diazepam whatsoever left in the channels. So based on that, I think we have a rough idea, and rough is the keyword here, that I think this is really helping us to start understanding at least these allosteric pathways. How is it that a molecule binding up here is within a microsecond or so changing the entire dynamics and the balance of the structure, modulated by binding sites down here, and it can force this allosteric modulators to be displaced, and then influencing the balance between the open and closed states of the actual pore. In the interest of time, I'm going to need to stop there, but there is no end to this. There are endogenous compounds such as neurosteroids, so in the nervous system, and the great thing here that there are so many channels that all of this could work on this for the rest of your lives, and we would still not be competing, which as a computational person is really awesome. And with that, I'd like to thank you very much for your attention. There are a number of heroes in Stockholm that we should thank at the end. And I'd be happy to take any questions you might have. Yep. There we have them. Thank you so much.
Thank you very much, Erik, for a fantastic lecture. I'm sure about that.
Yeah, maybe I missed something, but the sensitized states in your simulations, as a physiologist, those are highly stable states, and the dwell times are more in the tens of milliseconds, hundreds of milliseconds in the time scale. So that jumping out from the desensitized state take times.
Yes.
So how do you match with your simulation?
So there are some things that we can do in simulations that are difficult. For instance, when I remove an agonist, right, they're instantly removed. I don't have to wait for a perfusion system or something to gradually clear out things or so. That is likely one effect. The other answer is literally, I don't know. I would, if we get these timescales right within 1 order of magnitude, I would add, it's close to a millisecond. It might be 10 milliseconds too. And that it might also be that we're off by 1 order of magnitude. I don't know.
Thank you very much for the extremely interesting presentation. I have a question about the scheme that you showed. and what you call the intermediate and sleep states. As you may know, we had Pierre Jean and myself quite a lot of discussions about the so-called intermediate state that he had found as an independent kind of structure between the resting and active.
Yeah. You mean the locally closed or?
Now, well, the issue is that I just showed briefly a slide at the end of my presentation, is that we reach the possibility that, in fact, the so-called intermediate conformation is a way for the transition to go from the top to the bottom or from the inside to the outside. So that there would be, in fact, some kind of continuous transition, which would take place, if I may say, a polarity in the propagation of the conformation change through the protein, which is also in agreement with some electrophysiological measurements which have been. So I was wondering whether you capture this type of intermediate conformation here.
I think we do. There are some caveats here, right? In particular on the simulations, this is just one system, and we're still trying to get more sampling of this. That's why we haven't published it yet. But as you might have seen in this model, we definitely end up in this primed or fifth state or something that we call it. It's definitely we've had the extracellular domain move, right, and everything, and it's about to push the transmembrane domain open. But based on our classification, which again, these are not unique classifications, they're fairly low dimensional. But we definitely see things transiently moving back from the open partly down to this fifth state again. So yes, that I would argue that what we see is definitely a continuum, that we're gradually pushing it more towards the open state. So maybe starting by going between open and occasionally up to the fifth state, and then gradually we're pushing this so that we're moving between fifth and fully open. But yeah, the caveat here that this is just one sampling of one protein this far, so that, and we don't see it experimentally, of course.
Thank you very much. Thank you.
Yes, please.
Yeah, very nice talk. About benzodiazepine, Alicia's structure shows that there is no reorganization of the benzodiazepine site in the different states. So he was proposing that the benzodiazepine stick stability together, and this was the only role. So I don't think you're proposing a different mechanisms, if I understood well.
It depends on that. We definitely see a compaction of the transmembrane domain in the subunits, right? How much this actually corresponds to the pore opening is hard to say, that because in this case the simulations are not long enough that we're actually sampling. We don't sample any of the gating here. We can't see that we've made the open pore conformation more stable. So that there is a bit of hand waving involved here. No, I can't conclusively say that we've selectively stabilized the open state over the other one. In the extracellular domain, we definitely have the compaction around the binding site, and that's clearly associated with activation.
So it was not seen by CRYO-EM. Yeah.
No, not in the extracellular domain. We don't see the compaction.
I don't see the compaction.
No, not in the ECD, only in the TN.
Yes, please. Go ahead.
Very nice, welcome. My question regards the part where you say adding cholesterol stabilizes the growth conformation of the channel.
Desensitized.
Yes. My question is it because cholesterol binds at specific spots, at specific pockets, or because the physicochemical nature of the membrane is changed, or both?
I was about to say both. No, there are quite specific spots. It binds between the M3 and M4 helices. And there have been numerous groups starting to argue about the impact of the M4, the outermost helix in the structure. It definitely binds selectively there. And I didn't have time to mention it, but when we're adding this PNU modulator, PNU seems to displace cholesterol further up and pushes cholesterol down. And that might very well be that PNU primarily acts by preventing desensitization. And it might very well be that this displacement of cholesterol destabilizes the desensitized state. But yeah, I would say that. There's still a lot of argument about the exact role of cholesterol, but in these channels, I'm increasingly in the camp saying that we have quite specific binding sites.
Thank you for the talk. I have a question about this, where the lipid binding site to site 2 in the open conformation of the channel. So I was wondering if what types of conformation changes, what types of contractions you observe that changes so that there will be a lipid binding site appearing in the open conformation.
So there are many binders. So in general, the activation process of these channels starts out in the extracellular domain, right? And then you have this loop C closing in on the binding site due to the agonist being bound. That induces a slight rotation, in particular of the ECD relative to the TMD, that then pushes through the cysteine loop and then induces the rotation of the, well, this blooming motion that I think Jean-Pierre showed yesterday of the transmembrane domain. And I would argue it's not primarily that we open new binding sites. The binding sites are there all the time. In the transmembrane domain, I would argue even in the transmembrane domain, the binding sites are there all the time. The only question is that whether you selectively stabilizing the conformation of the binding site that corresponds more to the resting state or an open state or a desensitized state. But we never completely open new binding sites.
So maybe one last question about the Markov model that you showed for Glick, depending on the pH. I do understand your argument about sort of the proton being a partial agonist instead of a full agonist. But if you look at the barrier between the two states, you see it's 1.5 kcal per mole. So that's too small, right? So in MD simulation, you'll see reversible transitions with these barriers. How do you explain this?
Yeah, that's a good question. Remember, right, that it's even if under resting conditions, there isn't really any local minimum at the open states. Under resting conditions, you might move somewhat toward that direction, but you're never going to end up in any resting condition because it's never the lowest state. Under activating conditions, you're quite right. It's probably easier to somewhat go in that direction, but we still have this gigantic free energy minimum that corresponds to the closed state. So even if you briefly are moving in that direction of the open state, there is not any better free energy minimum there that would make the channels massively prefer to go in that direction. So even if you were to do, say, a shooting trajectories at the very edge here, in 90% of the cases, you're going to fall back in the closed states. So I think that the kinetics you end up getting, and that's of course why we end up getting these kinetics, that to actually open the channel will take you in the ballpark of 10 microseconds. Having said that, I don't believe that you have any accuracy here down to 1 kilojoule mole. That there is a barrier there. It's likely a few kilojoules or a few kcals. It might very well be 2-3 kcals instead. I have no idea. It's not 50 kcals though. But one should be a little bit careful about over-interpreting the quantitative results.
Projection.
Yeah.
Okay, thank you very much.
Thank you so much.